Dear students, the first lecture of histology is about the histological structure of the oral cavity, salivary glands and the tongue. But as you will see on the first two slides, I like to introduce very important terms related to the gross anatomy. In the last semester, you studied only two functions related to the nerves. One is the so-called somatomotor function. You can see this in red. When at the level of the spinal cord, the motor neurons are located in the so-called ventral horn, and their axons will leave through the ventral root, then they enter the spinal nerve, and then they travel either through the anterior or the posterior or dorsal ramus. And we have a direct contact with the target tissue, which is skeletal muscle, and we don't have any interruption in this path, so we don't have synapse, and the so-called effector of this function will be the motor end plate, what we study in the next semester. So this is labeled in red. This is how we innervate the skeletal muscles, and these are voluntary regulations. We have the so-called cutaneous innervation, what we studied in the last semester, the sensory innervation of the skin, which is labeled here with blue, and this is called somatosensory function or somatic afferent. In this case, the start points are the receptors, and the peripheral processes of the pseudoinoplan nerve cells will carry input from the receptors. Then without synapse, the info is transmitted to the so-called central process, which enters the CNS, and then we will study the termination in the next semester. So these are pseudoinoplan nerve cells, and the cell bodies are located at the level of the spinal cord, at least in the dorsal root ganglion. Now, these are the well-known functions, the somatomotor and somatosensory innervations. But in this semester, we study the splanchnic organs, the visceral organs, and that's why we have to mention the visceral innervations as well. We have both efferent or motor and afferent or sensory, but in this case we call them visceral efferent or visceromotor and visceral afferent or viscerosensory. A very important difference between the first one and the second, that in the second case, which is the visceromotor, we have always a synapse, we have always an interruption, so the efferent part is divided into two nerve cells. One is the preganglionic, labeled in black, and the other is the postganglionic, which is in green. And we will study later the site of the synapses. Can be variable, and we have some differences in case of the sympathetic and parasympathetic innervations. I don't want to go ahead. But at the end, we innervate smooth muscle or cardiac muscle, as you see, or glands. So this is how we regulate, not voluntarily, these target tissues, but always we have a synapse in this part, and the effector is called vegetative plexus. We studied this in the third semester. The corresponding sensory from the visceral organs, which are from the wall of the visceral organs or from the vessels, called viscerosensory, and I labeled it in blue again because we don't have difference in morphology between the somatosensory and viscerosensory innervations. In both cases, these are from receptors. The start points will be different. One is in the skin or in the striated muscle. This is the somatosensory. Or, in this case, from the wall of the internal organs, it means visceral organs or vessels. This is the interoceptors and viscerosensory function. And in both cases, the cell bodies are located at least at the level of the spinal cord in the dorsal root ganglion. But in your textbooks, you may find other subdivisions, and this is what I want to highlight to clarify this if it's possible. The first one, the somatomotor, has a special subdivision. Those skeletal muscles, which are derived from the so-called pharyngeal or bronchial arches, are innervated again with one direct nerve cell, as the first point is, but the name is different. This is called in your textbook bronchial efferent or specific visceromotor function. This is typical for these cranial nerves, what we study in this semester when we study the development of the pharyngeal arches and the derivatives of the pharyngeal arches. As you see here, these are related mostly to the face and neck related muscles, mastication, facial expression, and what we study in this semester, the pharyngeal and laryngeal muscles. But if we call this specific visceromotor in this case, then the previous one should be differentiated from this. And this is called general visceromotor in this case. But again, here we don't have synapse, here we have synapse, as we mentioned at the beginning. In case of the sensory or afferents, we may have also some subdivisions. First, I mentioned the somatosensory from the so-called exteroceptors or proprioceptors from the skin or from the striated muscles, respectively. And I mentioned the viscerosensory as well from the receptors of the visceral organs and vessels. These are called interoceptors. But we may have some very specific somatic afferent or somatosensory in case of the 2nd and 8th cranial nerves, so optic and acoustic infos are carried by this. And the so-called taste buds, what I mentioned very soon, the info is carried by the so-called special viscerosensory. And if it's called special viscerosensory, then the previous is called general viscerosensory, what I described previously. This is in general the interoceptor related, so baro, chemo, and osmoreceptor related. So we are able to detect the blood pressure, the gas content, and the osmotic pressure of the blood, for example. Now, after this macroscopic introduction, I like to start the histology. And in general, I can tell you that the epithelium of the oral cavity is a non-keratinized stratified squamous epithelium. We are not surprised because we mentioned in the first semester that where we enter from outside, from the skin into a canal, we have always a transition first, and this is called non-keratinized stratified squamous epithelium. Everybody remembers the layers. At the bottom, where we have a very mitogenic zone, that's why it's quite basophilic. This is the stratum basale, or germinative layer. Then most of the rows of the epithelial cells related to the stratum spinosum, or polygonal layer, where we have desmosomes, which make the cells spiky and spiny. And this will be seen only after a special histotechnical procedure. And on the top, we have a quite flat cell layer or rows of cells. This is called stratum planocellulare or planocellular layer. You see also, which is typical not only for the keratinized but also for the non-keratinized version, we see connective tissue papillae which provide a dual physiological role. One is the more stable connection to the underlying connective tissue. So this way, this is resistant to mechanic stimulus. And the other is the better blood supply, because with the way we transition between the epithelium and underlying connective tissue, we have more capillaries providing better blood supply. In general, the mucous membrane, I like to highlight it here, can be divided into two major layers. One is the epithelium, what I mentioned, and the other is the underlying connective tissue, which is called lamina propria. In case of the GI tract, we may have another layer, what we study later, a little smooth muscle layer, the muscularis mucosae. This is not seen in this picture. In general, we can say that the connective tissue is quite well vascularized. That's why the mucous membrane is reddish. Good wound healing is typical for the mucous membrane. And we may find, not in this photo, several glands in this. These can be serous or mucous glands. I like to highlight it. If you don't see keratinization, we won't see any gland from the skin. We don't see odoriferous, sweat, or sebaceous glands. It's a big mistake if you say this. Now, the first slide is about the lip. The outer surface of the lip is called cutaneous part, where we have stratified keratinized squamous epithelium with the regular, skin-like appearance. We may see hair follicles and associated glands. With this magnification, it's hard to see, but we see sebaceous glands. These are holocrine glands. And we see also sweat glands with merocrine secretion. The sweat glands have a special morphology, the coiled tubular glands, what we studied in the previous semester. Then we turn to the top. This is the so-called intermediate part, or red part. Here it's called rubor labii in Latin, or transitional zone. Here we have still stratified keratinized squamous epithelium, but very thin, and we have quite high connective tissue papillae, and that's why the capillaries in the underlying connective tissue are very close to the surface, and we are able to see the color of the blood in it. So that's why the red color will be provided by the blood. Here we don't have glands. Then we turn to the inner surface, and this is the so-called mucous part. This is where the mucous membrane starts. Here the epithelium is different. This is where we have stratified non-keratinized squamous epithelium, and we may find several glands, not in this photo, but these glands are already not the previously mentioned skin-related glands. These can be only mucous and serous glands, so salivary glands with merocrine secretion. The lip has several functional considerations. One is the fact that the intermediate part, or the red part, doesn't contain glands, and that's why easily desiccated, but we have special lipstick and tricks to wet it, to lubricate it. The other is the very sensitive, character of the lip. So we have several nerve endings and very well innervated gland. We will see that in the cerebral cortex we have a very large representation. We have a so-called oral phase in the newborn stage when we want to recognize our environment with our lip and tongue. You see this. And the other thing is that some diseases are related to the lip only. There's special type of the herpes virus infection, herpes labialis. In this case, the virus is settled in the ganglion, in case of the face, the trigeminal ganglion. And in some cases, when we have stress or some immune deficiency, it is flamed. The lip in the core has a very important skeletal muscle called Orbicularis oris. This is typical for this slide. And this is also very well innervated, and this is the other reason why we see several peripheral nerves in cross-section. And we are able to express our feeling or gestures, and you see here two shapes. One is kind of happiness, the other is rather frustrated mouth, and especially for the Hungarians, an important event was 2 years ago, the European Soccer Championship, when we were able to lead 3 times against Portugal. And, we were a bit lucky, honestly. This is our attacker, Balázs Zsuzsák, and the frustrated face, everybody knows him, Ronaldo. At the end, unfortunately, it was tied, but it's okay. The color of the lip is also very important and diagnostic. Normally, it expresses the beauty when it's red, and this is because of the underlying capillary network, as I mentioned too, because of the high connective tissue papillae in the red part. But in some cases, when we have hypoxia and low oxygen tension in the blood, because of the heart failures, for example, the color is rather bluish, and this is called cyanosis. And of course, we have to find the reason why, especially in newborns, this cyanosis occurs. In some cases, not the health problem, but the cold weather, the environmental temperature can cause this blue color because of the special defense against the heat loss. We have special circulation we study later, and vasoconstriction basically to skip the blood from the peripheral parts, and that's why it shows cyanosis in some cases. So you have to decide whether this is because of the cold weather or some more severe problem in the background. Now the next topic is the tongue, and what you see is that the so-called dorsal surface, which is rough, but we have another surface, the inferior surface, what you don't see here, that would be smooth. From a superior view, we see the dorsal surface only, which can be divided into two major parts with this V-shaped line called sulcus terminalis. It got the name because this was the proximal attachment point of the buccopharyngeal membrane, if you remember from embryology last semester. And that's why in front we have ectoderm derivatives regarding the epithelium and the glands, and behind we have endoderm. So behind we have endoderm. Let me highlight this point here behind this transition. This is the so-called foramen cecum, we will study very soon. This is from where the thyroid gland developed as the thyroglossus duct, and that's why the major cell of the thyroid gland, the so-called follicular cells, are endoderm derivatives. Okay, that is another story. Now let's see the gross anatomy first. The anterior 2/3 of the tongue is the so-called body or corpus. The posterior 1/3 is the root or radix. Histologically, the anterior 2/3 is the papillary part. The posterior 1/3 is the follicular part. What causes the roughness on the dorsal surface of the tongue? Anteriorly, the papillae, not the connective tissue, these are extra papillae, this is what I want to highlight. These are extra elevations. Of course, the connective tissue is elevated with the epithelial lining on the top, but these are extra elevations, and the original connective tissue papillae are much deeper, located deeper under this primary papilla. Again, we have epithelium lining on the surface, and we have the underlying connective tissue called lamina propria. So these are the two general layers of the mucous membrane. In contrast to the dorsal surface, where we see this roughness, on the inferior surface we see almost nothing special. We have in the midline the break of the tongue called frenulum, and on each side at the bottom we have a special elevation, a tiny protrusion called sublingual caruncula, favorite question in the exam, because this is where the submandibular duct and the major duct of the sublingual gland open. Otherwise smooth and mucous membrane covered area. Now back to the dorsal surface, to the body of the tongue, which is called histologically papillary part. It is made by the lingual papillae. We have 4 types of lingual papillae. One of them is just tactile papilla because of the mechanoreceptors related, and the other 3 have special role because they contain taste buds, and that's why these are called taste papillae. The filiform papillae, minute conical papillae, these tiny. The fungiform is a fungus-like. The circumvallate, we saw this previously, is the biggest one with quite a big ditch around. And we have the foliate papillae between 2 deep elevations. I mean depressions, sorry, leaf-like. The filiform papillae, as I told you, are minute, tiny papillae with keratinization at the top. This is very important for the animals especially, to be able to elevate and pick up and move the food in the oral cavity, so for feeding. If you watch this with scanning electron microscopy, as a rasper, tiny elevations are seen. In human, we may have also keratinization at the top, but in our slice, in some cases at least, they may be missing. So even if you don't see the keratinization at the top, if these are minute elevations, these are the filiform, and this will be between them the fungiform because these are randomly dispersed in the anterior 2/3. It shows a significant keratinization in this case. Of course, this is also highly innervated because these serve as mechanoreceptor. The fungiform papilla is much bigger elevation, and it may contain in the epithelium pale patches on the top or inner slides aside, but these are located in the epithelium, and these are called taste buds. And I like to highlight something. We have a significant difference between the connective tissue papillae and these taste buds. Both are pale in appearance under light microscopy, but in case of the connective tissue papillae, the whole pale patch should be surrounded by a basophilic layer because of the basal layer of the epithelium. But in this case, because it's a special organelle, let's say, we don't see this. Just a pale patch in the epithelium lining. So the fili and the fungiform papillae are randomly dispersed in the anterior two-thirds. It's able to see it in the mirror. And the fungiform papillae would be seen in larger red spots randomly. Now, I mentioned the circumvallate papillae in front of the sulcus terminalis in one row. These are the biggest papillae. We have 10, 12 papillae in front of the sulcus terminalis in the same orientation, in V shape. And you see large trenches aside. And here as well, as previously in the fungiform, in our case at least, we have taste buds, so pale patches in the epithelial lining. But what is very typical in case of the circumvallate or just vallate papillae, this huge amount of the secretory portion of salivary glands, because the ducts open in these trenches and able to wash out the chemicals from the receptors in the taste buds. So it's very important to be able to taste a new chemical. These glands are called, mostly serous glands, glands of von Ebner. The foliate papillae are located aside in the margin of the tongue. As I told you, one papilla is between two deep ditches, and these are secondary papillae then. And here you see again a relatively large amount of salivary glands. They open, even if you don't see the continuous connection, into these deep grooves. If we watch the taste bud, in Latin it's called Calliculi gustatorii. These are pale patches within the epithelium. Sometimes you see that there is a little pore to be able to be in contact with the surface of the groove or ditch. And we have basically two major cell types. One is the sensory cell or hair cell, which is quite elongated. You don't see the whole extension. But at the end, their processes are able to turn to the surface through this little pore. And we have oval nuclei, slender nuclei. They belong to the so-called supporting cells. We have four major taste types. We have the bitter in the root. We have sensation, in the root. We have a sweet sensation in the tip of the tongue. And laterally we have two types, the sour and the salty. So these are not related to the type of the papillae. This is what I want to highlight. And I added another type of the taste qualities, the so-called metallic and umami with the monosodium glutamate. I put it as advertisement, the famous Norwegian chocolate for the sweet sensation. Okay, what can we see in general in the slides? This is a very good opportunity when we see the lip slide to recap the basic structures what we studied in the first semester. So we see skeletal muscle in 3 dimensions. It's very important. This is the only organ in the body where the skeletal muscle fibers are oriented in 3 dimensions. What does it mean? We have 2 longitudinal skeletal muscle fiber orientation. One is vertical, let's say the other is horizontal, but longitudinal in both. And the third would be in cross-section. If we see this somewhere in the slide, we know that this is from the tongue, because this is the only organ where the skeletal muscle fibers are arranged so regularly. We are able to see serous and mucous glands, worth it to recap the major features of each. We may see ducts and lots of vessels. That is the good occasion to recap the vessels, because in the last semester the so-called vessel topic of the histo class was one of the last classes, and that's why you didn't have enough time to practice it, plus the COVID pandemic. And this is very well innervated organ, so that's why we are able to see lots of nerve cross sections. And we may see lymphatic structures, lymphatic follicles, or just lymphocytes, because this area is very close to the outer surface. It's not surprising that the body is prepared for the immune response with these lymphatic elements. And the root of the tongue is the posterior 1/3. Histologically it's called follicular part. Here we have the lingual tonsil. And here I have to introduce a special term, the so-called tonsil, which is a lymphatic organ. One of my colleagues, Balázs Gassner, will introduce the rest of the lymphatic organs. It's a very important lecture, especially now in the pandemic. The aggregation of lymphatic follicles in the mucous membrane. This is the definition of the tonsil, or covered by epithelium. But the epithelium should be there, and it's helpful to distinguish between the 4 types of tonsils, which are located otherwise in a special circle, what we study in gross anatomy as vallate lymphatic ring. In case of the root of the tongue, the epithelium is non-keratinized squamous. And we have another type of the tonsil in the oropharyngeal isthmus. We will study later this term, that is the outlet of the oral cavity towards the pharynx, where we have the palatine tonsil. These two have the same epithelial lining, stratified non-keratinized squamous epithelium. In advance, let me tell you the other 2 tonsils which are located in the epipharynx, in the airways, the tubarian tonsil and the pharyngeal tonsil, but not surprisingly, they have a pseudostratified kinociliated columnar epithelium, not this type. So we have 2 types of tonsils covered by this epithelium, and then we have to distinguish between the lingual and the palatine tonsil, and here I labeled 3 differences. One is the orientation of the skeletal muscle fibers. I just mentioned if we see them in 3Ds, then it should be from the tongue. That is the most crucial, because in the palatine tonsil, we won't see 3, only max 2 dimensions of the skeletal muscle fibers. The other difference between these 2 would be the amount of the salivary glands. In the lingual tonsil, we have much more mucous glands otherwise, and that's why this is the other reason why we don't have so many inflammations related to the lingual tonsil compared to the palatine. And but in the palatine we may see also glands, but less. And the third difference, that in this case, in the lingual tonsil, we don't see deep invaginations called crypts. This is typical for the palatine tonsil, and this is the other reason why the inflammations mostly will develop in the palatine. Now, this is gross anatomy again, but I like to highlight the innervation of the tongue because it's very important in this semester. And here I will use those terms what I introduced today, the so-called viscerosensory compared to the somatosensory innervation. The anterior 2/3 has a dual innervation. So the somatosensory, it means the mechanoreceptor, touch, pressure. Thermoreceptor, cold, warm. Nociceptor, pain. So these are in the sensory, in the somatosensory functions. This is related to the lingual nerve, which is a branch of the mandibular nerve, which is from the trigeminal. But in contrast to this, we have viscerosensory, the specific viscerosensory, if I want to specify this. And so the taste sensation. This is related to the chorda tympani, which is a branch of the facial nerve. But it's other story that the chorda tympani fibers first are carried by the lingual nerve. That's why it may cause difficulty in the next semester. But regarding the termination in the brainstem, we have 2 distinct nerves. One is for the somatosensory, the other is for the specific viscerosensory, for the taste. But in case of the posterior 1/3, the root of the tongue, we use only one nerve for both. So the glossopharyngeal nerve number 9 is able to carry both the somatosensory and the viscerosensory fibers, taste fibers. And here I like to highlight something. The taste sensation is not related only to the taste buds. What we mentioned in case of the papillae, these are all in front. Taste buds are seen without papillae in the root of the tongue, even more behind in the valleculae or around the laryngeal inlet. So we are able to feel taste behind the sulcus terminalis as well, but these are not related to the papillae. And here we use only one nerve for both somatosensory, for pain, let's say, and viscerous for taste, which is the glossopharyngeal nerve. And here I added another important thing. All the muscles of the tongue, extrinsic and intrinsic muscles of the tongue, are innervated by another nerve, the 12th cranial nerve, the hypoglossal nerve. Now, in general, we should turn to the salivary glands. In general, I can say that both the serous and mucous glands operate with merocrine secretion, and both, so if these are found in the big salivary glands such as parotid, submandibular and sublingual glands, what I want to describe, all of them are compound tubuloalveolar glands, so this way the most complicated morphology. Now let's see the differences again, but it's a kind of recap between the serous and mucous glands. The serous is much darker, basophilic, because of the RNA content. We studied in the previous semester that these cells have RNA-containing apparatus for producing proteins. These are the enzymes. Amylase, for example, so the carbohydrate digestion starts already in the oral cavity. Special defensive enzymes such as the lysozymes, we are able to break down the bacterial wall. Lactoferrin, which is able to bind the iron. This way the bacteria won't proliferate. So these are very important enzymes. These are proteins, and that's why we have RNA-containing apparatus in the cell, which makes it basophilic. We don't see lumen, basically. Very narrow lumen, even not visible. And the third, that the nuclei are relatively regularly located, rounded in the cells. If we compare this with the mucus, the color is very pale because of the dissolved mucin, and we have much more visible lumen, and usually the nuclei are compressed to the bottom. This type of the saliva produces rather, or gland produces rather slimy and sticky saliva. Let me stop here for a second to express the serous demilon, the Januzzi demilon, what we studied. I studied myself when I was a student, but it seems an artifact now. After a special histotechnical procedure, the mucous glands is swollen and pushes out the rest of the serous to the periphery, so it's not a kind of transition as previously we believed. Under electron microscopy, you see the two different glandular types. The serous cell contains tiny electrodense, so dark vesicles. The mucus contains paler but larger vesicles. In general, we can say that in addition to these salivary gland secretory portions, which are located in the lobules, these are separated by connective tissue septae. This is where otherwise the vessels, the nerves travel, and the large ducts as well. But before I go to the details and to distinguish the different big salivary glands, I'd like to show you something new, because in the previous semester it was enough to recognize the duct compared to the secretory portions of the salivary glands. But in this case you will see that we have different duct types. At the beginning, as the continuation of the secretory portions, we have a so-called intercalated duct, what I labeled in blue here, and this is a simple cubital epithelial lining. Then it continues in the green. This is the so-called striated duct, which has a columnar appearance, simple columnar epithelial lining. Both ducts are found still within the lobule. These are called this way intralobular ducts. But if you look at the green, with larger magnification on this schematic drawing, you see that at the bottom we see mitochondria accumulation. Between them we have invagination of the basal cell membrane, and this phenomenon is called basal striation, basal striation because it is on the bottom of the cell. This way, from the mitochondria, we are able to gain more ATP, which is required for the pumps, which are located in the apical portion, where we have the striated border in some cases. And this way, we are able to change the ion content of the saliva. So these two types of the ducts are found in the so-called lobule, and that's why these are called interlobular ducts. But we have a big one, the so-called excretory duct, which is found outside the lobule, and this may have even pseudostratified epithelial lining. So let's see this again. You see serous and mucous secretory portions, and between them, inserted, we have a little pinkish structure with simple cubical epithelial lining. This is the very first part of the duct system, so the intercalated duct, one of the interlobular types. And if it becomes columnar, then it is striated, and if we see them outside the lobules, between the lobules in the interlobular connective tissue, these are called interlobular ducts or excretory duct. The striated duct, so this is what I wanted to tell again, or a basal striation, is made by the invagination of the cell membrane. You see these duplicates of the cell membrane under electron microscopy, and between them in the cytoplasm we have accumulation of mitochondria. This is important for the ATP generation, which is required for the pumps in the other side of the cell in the so-called apical portion. So that's why they are able to make a sodium-potassium exchange. Now, if we regard the big salivary glands, we have three big ones, the parotid, the submandibular, and the sublingual. The proportion of these two types of secretory portions will be the crucial. The morphology otherwise everywhere is compound tubuloalveolar glands, and the mechanism, how they operate, always merocrine. But the proportion will be different. In case of the parotid, we have only serous. In case of the submandibular, we have mixed, but with serous dominancy. That's why I used capitals for this. And in case of the sublingual, which is located under the tongue, we have again mixed, but with mucous dominance. Here, let me highlight immediately that in the third one, we have much less ducts in cross-section. This is also typical in the slides. Now, if we see the photos, the parotid is this because we see only serous secretory portions. Be careful with these pale patches. These are not mucous salivary glands. These are dissolved fat cells. And we see quite well elaborated duct system within the lobule. So this is the parotid. In case of the submandibular, we have already mixed appearance, but with serous dominancy, less mucus, and we have again well-developed duct system with high magnification. This is the serous, this is mucous gland, secretory portion, and these are the ducts. In this case, these are intercalated ducts. You see the difference. This is relatively bluish, this is pink, the regular color of the cytoplasm. And the third would be the so-called sublingual gland, where we have more mucous secretory portion, much less serous. And again, I like to highlight, we have much less duct, and that's why you don't see these in the photos. If we use a special staining method for the visualization of the carbohydrates, the pus, it's able to visualize it, and they will appear in purple. And let me highlight this. In this photo, you see the accumulation of cell bodies of nerve cells in the periphery, so this will be a ganglion. This can be only vegetative ganglion if we refer to the previous semester topics. Even if you don't see the processes of the nerve cells, with hematoxylin we are not able to see, but here we are able to see only vegetative ganglion, and we see two cross-sections of peripheral nerves. And at the end, I'd like to show you some statements about the saliva. The daily production is 1.5 liter, and mostly the submandibular gland produces this 70%. The rest is shared between the parotid and sublingual with parotid dominancy. And composition, we see electrolytes, and we have a so-called primary saliva after the production of the saliva in the secretory portion, but then when it passes through the duct system, as I told you, the striated ducts are able to modify the ion content. Less sodium will be at the end, and that's why hyposmolar will be. This is the typical pH range. And then some organic components are listed, proteoglycans and glucoproteins, that's why it's a mucous component. And we have important enzymes, as I mentioned, amylase for carbohydrate digestion, lysozyme and lactoferrin for defense against bacteria. And we have a special immunoglobulin. This is typical for the fluids in the body, so such as the saliva. This is produced by the plasma cells, as we studied in the previous semester. But this immunoglobulin is typical here, the immunoglobulin A, as in the other fluids in the body. And we have some other plasma proteins. And at the end, we have dual regulation. Parasympathetic and sympathetic, both are vegetative or autonomic. The parasympathetic innervation will induce the enzyme-rich saliva, which is water-containing, watery. And we have the other one, in case of stress, for example, we have more slimy, more viscous saliva, and this is made by the sympathetic innervation. Okay, you will see these slides much better in the regular class, which will be hopefully in the seminar rooms in this semester. Thank you very much for your attention.